Hello students, welcome to Vedantu Neet English. I am Miss Gopika, your biology master teacher. How are you all doing? Let me know in the chats how all of you are doing, and if you are able to see me, hear me, even also see the presentation behind me. Okay, are we live? Tell me fast. Are we live? No, we are not yet live. Yes? Let's wait for the... Come on students, let me know if I'm audible, visible. And also we will check, right? Yes, we are live. I am live. Are you able to see me? Yes, now ad is playing. After that, I'll be live. Okay, so today we are going to do biological classification, right? So this chapter, usually what students do it, first chapter, no? Like after living world only little is there. Biological classification, all the students will study properly. Okay, after that, when they come to plant kingdom, when they come to animal kingdom, they lose the interest. They'll be like, oh, again I have to study a lot about this, sir. So biological classification is a type of chapter that students will be like full energy. Today I am going to crack Neet 2025, 2024, 3, whatever, how many, whichever years you wrote, all of you will open your book and be like full dedication. So are you also here for that dedication? Yes or no? Yes, good evening all of you. How are you all doing? And did you smash the like button? Did you mark the attendance? Why will you give me bad name like attendance ma'am and all? It's not even nice to hear. Okay, so today we are going to do biological classification, students. Last year, my 2024 students, I would have told my students it's okay, last minute you revise this chapter, or last minute you do the living world, microbes chapter. But this year, since everything is gone ulta pulta, right, with the whole Neet, I think we shouldn't skip any chapter. Even if it's giving us 4 marks, we should never skip the chapter. Yes or no? Do you all agree with it? Yes, awesome, very good. All of you are putting fire emoji without me telling. Very nice, very nice. You people have improved in life. Okay, so yes, what are we going to do, students, today? So since the chapter is very long, I will be not doing the entire chapter. I'll be splitting it into 2 parts and doing the chapter. Okay, first part is today and the next part will come very soon. Okay. So what am I going to do? I'm going to do the introduction. That means I will teach you about all the kingdom classifications. What was who did the classification, which was the date, what was the drawbacks, all of it. Second thing is I'm going to talk specially about 2 kingdom classification, because according to your NCERT, you should know 2 kingdom classification. Okay, chalo. Then 5 kingdom classification, your favorite classification, Whittaker's classification, right? Yes, okay. Then kingdom Monera. Kingdom Monera, after that you only have Protista and Fungi, which I'll finish it in one class. And then Plantae and Animal, by itself they have given a kingdom, Plant Kingdom and Animal Kingdom, right? Okay, yes, NCERT, by your new NCERT, okay? Now, students, are we ready? All full energetic, full Josh is there, Did you all download the last notes that I sent you? Let me see the attendance first. First attendance, then talking. Okay? First your job you have to do, then only I'll do my job. Chalo, let me see. Where is the attendance? Now scoldings will come. Okay? Scoldings will come. Now on scoldings only for you people. Now you already know me, from 1 year you know me. So now on scoldings for all of you. Okay? No more telling, oh please do it. No. Scoldings. Okay? Yes. Now let's start. So students, biological classification, before we get directly into kingdom Monera, Protista, we have to understand how was the first classification done. The first classification was done with no scientific validity. That means basically I did a classification with no scientific evidence. Okay, no scientific evidence. I'll be writing every single thing. Okay, I'll be writing every single thing like you want. My notes is there. You will get it on Vedantu Neet English. Okay, and you can also follow me on my Telegram as well. Okay, thank you, Rama. So students, with no scientific classification, they did first classification that was based on food, shelter and clothing. What do I mean by this? That means with no scientific validity, I just classified organism. I classified your entire ecosystem, your fauna, flora into whoever gives me food, I'll put them into food. Whoever gives me shelter, I'll put them into that. Whoever gives me clothing, I'll put them into that. Can you do a classification like that? All of you tell me, you're all science students. Do you think this classification is appropriate for me to take it forward? Definitely not, because I cannot classify organism depending on my personal basic needs. My selfishness. My selfishness is that I want to get food, I want shelter, I want clothing. I did a classification. So this did not go well. So the next classification that came into existence, or let's say the first ever scientific classification, okay, first scientific classification. I want all of you to write notes with me. I know you will get these notes, but at least scribble something and write it in your NCERT as well. Okay, first ever scientific classification was given by who? Any of you last year, my students? Anyone who is here? Anyone who is there? Subhashini, give me the answer. Pratibha, Trisha, Pavitra. Come on, soul of Indian mythology. Chota name you keep so that I can call you very oftenly. Okay, so what is that? First ever scientific classification was given by Aristotle. Who? Aristotle. He was the first person to do a basic classification. I'm not telling this was the best classification, but I'm telling you he was the first person to give at least some scientific approach to it. Okay, what was the scientific approach he would have taken? The simplest scientific approach he would have taken is to divide them into plants and animals. Simple, students, even you could have done it. So simple classification that they did was he divided the entire organism into plants and animals. Now, on what basis he did the classification? We have to know if it's a hit or pass, right? So he basically classified into two things. Okay, first he divided plants, okay, plants and animals. Now, how was this classification continued? That we have to see. Plants were categorized based on their habitat or based on their sizes also, we can tell. That is, he divided plants into three categories. What is the category? You have studied it in your EVS. Herbs, shrubs and trees. So basically the ones which are small, I will call it herbs. Then a little more bushy ones, I will call it shrubs. Then I will directly jump into trees. Okay, this was the classification. Okay, but it has some scientific validity, right? At least I put them into plants. At least I categorize them into animals. Now, he put animals. He did the classification of animals based on the presence and absence of RBC. Okay, presence and absence of RBC. I'm writing it here for you to understand. Okay, presence and absence of who? Of your red blood cells. That means depending on the blood color. Okay, what pigment is there? Is it red in color? Is it white in color? Is it colorless? He divided them also. Okay, he divided them also. That is, he divided animals into two categories. Okay, he divided them into, no space. 1 second, students. Presence and absence. Okay, RBC. Okay, sometimes I end up writing RCB. If you see some of my notes, I end up writing RCB. Okay, because too much RCB is in my head. Very easy, no? RBC, RCB. So I end up writing presence and absence of RCB. Okay, so here it is RBC. So they divided it into enyma, enyma and enyma. Okay, like two siblings. A stands for absence. So enyma means absence, enyma means present. Is this clear? Okay, is this clear? So this was the classification. The first ever scientific classification was given by who? Students, the quiz, the PYQ will not come. Oh, why did he divide it into herbs, shrubs and trees? The question will come in such way that who was the first scientist to do the first ever scientific classification? All of you tell me in the chats. Who was the first person to do the first ever scientific classification? That will be, answer should be Aristotle. Okay, they will not ask you this easy questions. They will ask you little twisted questions only. Now they'll also ask you enyma. What does that word stands for? That means you have to tell enyma stands for presence of RBC. Enyma stands for absence of RBC. Good, good to go. Okay, Aristotle, gave respect to Aristotle. Yes, ma'am. Okay, done. Fine. Now we'll come into a classification that made little more sense. Like students, what is basically happening is, now Trisha is, okay, wait 1 second. Okay, Trisha is doing this two kingdom classification with no signs. So Subhashini is watching from far and Subhashini is like, hey, what is this Trisha doing? I can do better. Like that, when scientists came as competition with each other, they were watching the other person from far. So one person came with 2 kingdom classification, other person came with 3 kingdom, 4 kingdom, 5 kingdom, 6 kingdom classification. Every kingdom is a improvement. Okay, every kingdom is a improvement. Got it? So now the next person who came up with some more valid, points about the classification was your Carl Linnaeus. Where have you studied the name Carl Linnaeus? All of you tell me in the chat box. Where have you studied the name Carl Linnaeus? Come on, tell me. Yes. Yes, enema. That's what I told you, in the presence and absence of RBC. Some people also tell it. 1 second, students. Yes. Tell me, students. Nomenclature. Very good. Is my voice audible? Voice audible? Yes. Okay, binomial nomenclature. Correct? Okay. Now, this was the first fellow to tell that, okay, there's more science to it. So, Carl Linnaeus, okay, Carl Linnaeus was the person. Students, don't tell me, ma'am, you did not underline and all, because when I'm writing suddenly, I will not be able to underline everything and do properly. So just understand, okay? I know nomenclature. So yes, Carl Linnaeus came up with the actual two-kingdom classification, okay? I know here also we did a classification, but again, this made more sense. So we call this as the first two-kingdom classification, okay? What do we call? Two-kingdom classification. Two-kingdom classification. Now, what is this classification what's so special about this classification? As I told you, some more science was added into this. Now, what was the science? He basically classified everything on the presence and if they have cell wall or if they do not have cell wall. Yes, if they have cell wall and if they do not have cell wall. Is this clear to all of you? Yes or no? Presence and absence of cell wall. Okay, did you all like? Come on, students. Put the attendance. As you join, you should mark your attendance. Then only I know that, okay, the student was there in my class or this many students were there. Okay. So, two kingdom classification, he started on very simple basis, that is presence and absence of cell wall. I'm not telling this was the ultimate of it. Okay. Cell wall. Again, simple classification, plants and animals. Okay. Plants and animals. But one twist is that he did not put everything properly, okay? He did here and there some gut-butt problems he did. That is, he added so many things. Students, I'm going to write some of it, okay? He added dinoflagellates, okay? He added dinoflagellates. He added diatoms, okay? He added bryophytes. Now again, you don't have to remember everything, but if you can remember, well and good. He put pteridophytes. Okay, he put pteridophytes. He put angiosperm, etc, etc, etc. He kept adding many things. Okay, diatoms, dinoflagellate, cryophytes. Then he put bryophytes, pteridophyte, angiosperms. In animals also, he did some things like he put like amoeba. Okay, he put amoeba. He put paramecium. Can you tell me why he put amoeba and paramecium here? And he also put other animals. Okay, he put rest of the animals. Can anyone tell me why he would have put amoeba and paramecium? Yes, tell me. Yes, Savita, we are going to come into that, okay? We are going to come into that, okay? We are going to come into based on cell structure, mode of nutrition and all that we are going to come, okay? Many things were missing. Okay, many things were missing, but the basis he did was based on the cell wall, presence and absence of cell wall. Again, many things did not come into place. Okay, we are going to see what are the drawbacks as well. Now, you tell me, why do you think he would have put amoeba, paramecium with all the animals? Can anyone tell me? Anyone tell me? Absence of cell wall, yes, that is a main category, but he also told something else. Yes, they have no cell wall. Yes, correct. But he also, don't you think he put them this here maybe because of slight locomotion that they're doing, some movements, right? Whenever we talk about animals, what do we tell? Okay, they're able to do something, some kind of movement, right? So that movement also would be the reason why he put it under animals. Again, there are multiple people talking about multiple theories, okay? Some people tell more things. See, I generally told presence and absence of cell wall because again, that is what is mentioned in your NCERT. But many other people tell more things. Okay, cell structure was taken into place, that was taken into place. So these theories keep going around. Because Carl Linnaeus is not here, so anyone can write anything, right? So that is the whole thing. So is this clear? Is this clear? Two kingdom classification is clear? Let me know in the chats. Two kingdom classification is clear or not? Okay. Animals, plants, and he added most of the plants, multicellular, unicellular, all of this into place. Now, the catch is, what was the problem? What was the problem? Students, I'll write here, okay. Okay, I'll write here only. Yes, what was the drawback? I saw bryophytes with dinoflagellates. I saw bryophytes with other blue-green algae. What is the main issue? The issue was that he did not do proper classification. So we are going to understand what are the drawbacks. Okay, what are the drawbacks? You might be thinking, ma'am, first of all, that thing failed. Why should I study the drawback? It is to understand from where the other scientists build their whole thing. Students, if there are no mistakes, in NEET 2024, whatever mistakes you did, that you will not repeat in NEET 2025. Agreed or disagreed? Agreed or disagreed? Okay? Yes, yes, definitely. DNA batch can see, YouTube batch can see, everyone can see, students. No issues, no issues. Okay? So listen here. If you know what mistakes you did this year, you will not repeat it next year. So drawbacks are very important. So drawbacks of your two kingdom class. Students, please understand, whenever I am telling two kingdom classification, I am talking about Carl Linnaeus classification, not about Aristotle's classification. Many students end up thinking that whenever we are talking about two kingdom classification, it is about Aristotle. Okay, that much respect is enough. Okay. So much if you think about that person, you will end up making mistake. Okay, what were the drawbacks? First thing I noticed, what are the things that you noticed? I noticed that he did not classify prokaryotes and eukaryotes. Yes or no? He did not classify prokaryotes. Okay, prokaryotes and eukaryotes. How do I write it? Okay, I'll write prokaryotes with eukaryotes. Okay. I'm not completing the statements, but yeah, I'm just adding the important words that you need to remember. Okay. The next thing he did not bother much was about very simple thing, unicellular and multicellular. Yes or no? Unicellular and multicellular. Multicellular. He did not bother. He was like, okay, you all come under plants. Okay, multicellular. Next thing he did not bother much was, students, photosynthetic and non-photosynthetic. Okay, fungi also was there. See, I haven't written all of it. Okay, if you write all of it, you know that many people were in this place. Okay, fungi were there, your blue-green algae were there who do photosynthesis and who are heterotrophic, or who do autotrophism and heterotrophism. So here, the next thing that he did not bother much was photosynthetic and non-photosynthetic. Photosynthetic and non-photosynthetic. Okay, and non-photosynthetic. Again, these are the only keywords that you need to know. Okay, don't worry, I'm writing every word of NCERT. You do not have to take tension. Okay, I am writing and teaching because you have to write and learn. Without that, there's no other option. Yes, very good. Unicellular, multicellular, photosynthetic, so very good. All of you are answering. Awesome. Okay, what is the other thing? Now, since I told you photo and non-photo, then you should also tell autotrophs and heterotrophs, right? You should also tell autotrophs were put together with heterotrophs, right? Autotrophic and heterotrophic also were not categorized between each other. Okay, heterotrophic were not categorized. Other than that, what did you see, students? Anything else you people saw? Something else you people saw? Unicellular, multicellular, prokaryotes, eukaryotes. So he did not do proper differentiation, right? No differentiation was made, right? I'm writing it here because there I couldn't write. Okay, no differentiation was made. Students, and also let me tell you, there were people like slime molds who were not put into plants also, who were not put into animals also. Now what to do about them? Can I take and throw them? Like, hey, you don't have place here, go. So some of them did not belong to plants and animals also. That was also a problem. Okay, so I'll write here, some of them did not belong into plants and animals at all. Okay, some were left out. Okay, some what do I write? Some, I'm thinking what to frame it well. Okay, some organisms belong to plants and animals. Okay, some did not belong to plants and animals. Clear? These were the drawbacks of Carl Linnaeus. Now, I told you, Carl Linnaeus is doing mistake, someone else is watching him. Okay, so when you are doing a mistake, someone is always watching you. Yes or no? Yes or no? Chalo, tell me, all of you. Oh, what is the students' chat summary and all is coming? YouTube has fully upgraded themselves, huh? Upgraded themselves. Okay, chalo, let's do it. So, this much clear? So far good. Now, students, I told you, when one person is making mistake, another person is always active. So then we thought, okay, at least let's rectify some of the issues. See, we cannot rectify everything and put them all suddenly into categories. One by one, slowly we have to get into it. So let's see who are the other people who got into it. Okay, who are the other people who got into it? So I have made a box entirely. I'll also make you write it, like what were the difference. But as of now, okay, just try to understand. So students, Carl Linnaeus in 1735 put Plantae and Animalia, okay, or your plants and animals. Under that he categorized many things. Whatever drawbacks he did, what happened? Ernest Haeckel, okay, a scientist called, you do not have to remember the dates of everything, you will have to remember only R.H. Whittaker's date, okay, only R.H. Whittaker, that means when 5 kingdom classification came into place, that's only what you have to tell, okay. So here Ernest Haeckel in 1866 categorized Plantae Animalia. Then he told, okay, let's do one thing. The main problem that you have is about the unicellular and multicellular. Let's do one thing. Let's put all the unicellular into one place. So he put all the unicellular into Protista. So who was the person who came up with Protista? Your Ernest Haeckel. You have to know the names, okay. They will see in NCERT it is not there, but they know that when a person is preparing you for NEET, they'll teach you this. So Protista, what is the thing? He told, let's put all unicellular into that. Okay, all unicellular into this. Now, what do you see the drawback? How can I put all the unicellular prokaryotes and eukaryotes into one? Then another person came. Okay, the another person name was Copeland. Okay, Copeland came. Students, Ernest Haeckel will also study in evolution. When we study your embryological evidence, you'll be talking about Ernest Haeckel. Okay, so give nice respect and all. Again, we have to learn about him. Now, Copeland came and he told, let's do one thing. Let's put all unicellular prokaryotes into Monera. So what did he tell? Let's put all unicellular prokaryotes into this category. Okay, into this category. So where will eukaryotes go? All of you tell me in the chat box, where will eukaryotes go? If prokaryotes has been put into Monera, all eukaryotes will go into Protista. Okay, so U and Pro we separated, Uni and Multi also we will separate slowly, or depending on U and Pro, let there be some here and there exchange, because again you know prokaryotes, unicellular and multicellular also slowly got differentiated. Okay, now so Monera, Protista, Plantae, Animalia. Now comes the most renowned, most accepted classification, that is called as the 5 kingdom classification by R.H. Whittaker. You have to know the date, that is 1969. Okay, 1969. Reverse letters, okay. 1969, that is he came up with fungi, because students understood here also when we put this, fungi is not belonging anywhere, because you know fungi by itself is so totally different. So fungi was not belonging anywhere. So R.H. Whittaker was like, okay, let's not break our head, let's give one full kingdom to fungi. So they gave entire kingdom to fungi. Then after that also there is Carl Woese who came in 1990. Okay, and he told, let's do one thing, let's divide it into 3 domains and 6 kingdoms. So all of you know above kingdom is domain. So he categorized them into domains. I'm going to teach you that also. Now, who and all came? Can you think and tell me who are the scientists who gave each classification? Okay, let's do one thing. Let's split the board. Okay, you have to tell me who did what. Okay, who did what? Okay, let's start with Ernest Haeckel. I think the chats are loading a little late. Okay, Ernest Haeckel. What did he come up with? He came up with, wait one second. Ernest Haeckel, he came up with what? Yes, I think here chats will be faster. Yeah, tell me fast. Ernest Haeckel came up with which name? Protista and Monera. Most of the students, what they do, you know students, they write See, I did not write. Okay, they write Protista. They write Monera. They think Monera. After Monera, Protista came. So Ernest Haeckel came up with Protista. Okay, Protista. In this, what was the plan? To put all unicellular together. Okay, all unicellular together. Students, you have to type fast. Come on, you have to type fast. Next is Copeland. Copeland came and he was like, hey, how can you put all the unicellular together? What about U and Pro? So Copeland came with Monera. Copeland came with Monera and he told, let's do one thing. Let's put all. Students, you have to know in each category who went. Okay. Like when I separated them, who went into each category. All unicellular prokaryotes, let's put it into, okay, let's put them into your Monera. Clear or not? Easy peasy, lemon squeezy? Yes. The next person who came was the most important person, that is your R.H. Whittaker. Okay, R.H. Whittaker. He took everything and went, Whittaker. Okay, he took the whole name for the classification that is called as your 5 kingdom classification. So he came up with the 5 kingdom classification. Please understand, even though 6 kingdom classification is existing and it came after 5 kingdom classification, still the most widely accepted one was R.H. Whittaker's 5 kingdom classification. The question can come, which is the most accepted classification? That is your 5 kingdom classification. 5 kingdom classification, the main change that he made was to add a whole, give a whole kingdom to fungi. Okay, when you know that they are a problematic one, you just push them out into another kingdom by itself. Next kingdom, okay, the next one was 6 kingdom classification. Now in 6 kingdom classification, you need to know little. Okay, 6 kingdom classification was given by Carl Woese. Okay, Carl Woese. Now students, that is not important thing. The most important thing is, what is this domains, ma'am? Right, he divided them into how many domains? 3 domains. 3 domains. Now what is this 3 domains? You have to use a little, you should try to understand how did he do this. So you should know kingdom is above domain, right? Kingdom is above, no, domain is above. Domain is above kingdom. Kingdom is here, students, domain is above. Okay. So he basically divided them into Archaea, Eubacteria, Eubacteria and Eukarya. That means eukaryotes, no? Like that, eukarya. This was the 3 domain that he divided it. Under 3 domains, he told, let's do one thing. Okay, let's do one thing. Under the 3 domains, let's put the kingdom. So he told 3 domain and 6 kingdom. Now, you tell me Archaebacteria. Okay, have you heard that name? Yes, ma'am, you have heard that name. Okay. Yes, what is Gopal telling? Monera into 2 and remaining as 1. What? Okay. Yes. Yes, Carl Woese. Okay, tell me students, what was Carl Woese's whole logic? He was like, chalo, I will do one thing. I will put under Archaea, I will put Archaebacteria. Okay, under, because Archaebacteria, you know, it can withstand extreme conditions, and again, sole members are bacteria. Under Eubacteria, obviously by the name only, you know, it's Eubacterias. Okay. Eubacterias or the true bacterias. I basically divided into the false bacteria and the true bacteria types. And eukaryotes. Who are the eukaryotes? Definitely the rest of the 4 of them. Okay, who is going to come into this? So who are the 4? Protista. Okay, Protista, fungi, plantae. Okay, plantae and animalia. Simple as that. Simple as that. Yes or no? Yes, yes, very good, Savita. Okay, so this was the classification. So understood, 3 domains, 6 kingdoms. Please do not write 3 kingdoms, 6 domains. Okay, right? 3 domains, 6 kingdoms. Okay, so this was the whole category with which he did the classification. So in 6 kingdom classification, if I have to write it this side, 6 kingdom classification. Okay, let's write Carl Woese. No, should give. Okay, Carl Woese. What did he do? I'm writing it here. 3 domains. Students, you read my notes. After learning my notes, you read NCERT. More than enough. You do not have to follow 100 other books. Okay, read my notes because I am reading NCERT and making these notes. So I will not miss out on anything. Even if I miss out on something, I will add the NCERT snip as well. So read my notes, after that read NCERT. That's why I'm making it this colorful, otherwise I would have written it in one color. Okay, so Carl Woese came up with 3 domains and 6 kingdoms. So utilize the notes, students. You're getting it freely. Please utilize it. Okay, done. Okay, this is also clear. Now students, R.H. Whittaker did a good job for all of you. R.H. Whittaker was like, my kids, don't study a lot about me. Okay, one box I'll give you, you study only from that box. Okay, students, only this box you have to study for R.H. Whittaker's entire 5 kingdom classification. From this box only, multiple different types of questions every single year, it will come from this box. That's why every teacher stresses on this box a lot. Okay, NCERT, yes, NCERT is enough for Neet. This last year also we saw how NCERT is very, very important. Now students, look here. What is the classification based on? Very simple thing. The classification was based on cell type, cell wall, nuclear membrane or body organization, mode of nutrition. Okay, so nuclear membrane and then body organization. So basically R.H. Whittaker did not do a blind approach. He did not be like, okay, Copeland put protista, so I will Chuma come and put fungi. No. Okay, what he was trying to say was, he was trying to say that I will do a classification on such a basis that all the organisms will come into it and no one will be able to question it. That is, I am going to classify it based on cell's wall, cell structure, body organization, nuclear membrane and mode of nutrition. Now more than this, where will you get? Students, you will get my notes on Vedantu Neet English Telegram. The link is right below. Okay, below you can see the Vedantu Neet English Telegram. Got it? Yes. Okay, look here. Here students, Monera. All of you know Monera is prokaryotes. That's why we put all unicellular prokaryotes into Monera. After that, everything is eukaryotes. Okay, everything is eukaryotes. That is your plant from your protista till your animalia is the same. Now, cell wall. Non-cellulosic polysaccharide plus amino acid. Instead of this, you can simply write peptidoglycan. What you can write? Peptidoglycan. Oh God, no place here students. I'll write it this side, okay? I'm writing it here. Okay, peptidoglycan. That means your polysaccharide, that is your sugar plus amino acid together will form your peptidoglycan. Is this clear or not? Okay, red is visible, no? Should I write it with black? It's visible, I guess. Okay, peptidoglycan cell wall. Okay, that is polysaccharide plus amino acid. Now, present in some cell wall. We are talking about cell wall. In protista, it is present in some, absent in some. Next class, when we study protista, you will understand this. Now, chitin. That is here in fungi, it is chitin. In fungi, it is chitin. That is simply, it is non-cellulosic. Okay, non-cellulosic. The question will not come directly as chitin. The question will be, which is the kingdom that has a non-cellulosic cell wall? So your answer should be in that way, non-cellulosic. You know only plants have cellulosic cell wall, rest all of them has non-cellulosic cell wall. Okay, again in protista also if some exceptions are there you can leave, but generally if the question comes you should know which are the ones, and absent in animals. Now nuclear membrane students, definitely because this is prokaryotic it will be absent, in everything else it will be present. Body organization, look here, cellular. So the question will be, which is the first kingdom to have multicellular body? That was first was fungi, and they also have loose tissues. That is, you know, mycelium hyphae formation. Again, I'll teach you all of that. Loose tissues were formed. Okay. Now here also tissue organ level, tissue organ system. So the questions I'm going to ask you rapid fire after this. Okay. After this box, I'll ask you a question. Got it? Okay. Yes. Now, mode of nutrition is autotrophic. See, here, what is the most catchiest thing? Okay, most catchiest thing. Non-cellulosic, dear, just non-cellulosic. Your chitin also comes under non-cellulosic. That means it's not made up of cellulose. Okay, that's all is the question. Non-cellulosic. Okay, yes. Now, look here. your Monera is the only one, only kingdom that can do chemosynthetic autotrophism. That means use chemicals to make their own food. Under autotrophs, they can do chemosynthetic as well as photosynthetic. That means with the presence of light also they can do, with the presence of chemicals also they can do. Please try to understand, the students, please try to understand. Understand, under autotrophic, chemo and photo is coming. It's not like separate, okay? So this is the only one that can do chemosynthetic autotrophism, and again photosynthetic heterotrophs also there. Here also autotrophs and heterotrophs, that is your protista. Here heterotrophs, saprophytic and parasitic. Fungi, you always know, again that is saprophytic and parasitic. Again your plantae autotrophic, but again exceptions are there, like your cuscuta and your insectivorous plant, like your Venus flytrap, pitcher plant, all of that are exceptions, okay? They are parasitic in nature, okay? Parasitic in nature. Got it? Any doubts so far? Any doubts? And here you have Animalia, that is heterotrophic holozoa. That means first we ingest the food, then after that digestion happens. That means because of the enzyme, the food gets digested, okay? Yes, students, drink water, all of you. Yes. Yes. Okay. New students haven't marked their attendance, okay? If you don't mark attendance, you don't get blessings. Understood? Serious issue it is. Okay, this is clear. Shall I ask you questions? Okay, look at this. Look at the box only and answer. The first question to all of you. I think the chats are slow here. So here, I'm checking here. First question to all of you. Tell me which was the first ever kingdom to have, okay, to have multicellular organization. The first ever kingdom to have multicellular organization. You have to be quick, okay? You have to be quick. Chalo, tell me. No, Priya, chapter will be completed in 2 parts, dear. 2 parts. Now we can do it, okay? Little aram se we can do it. After some time speed will increase, okay? Very good. Fungi is the answer. Good job. Now the next question is the one which has chitin as the cell wall, okay? Chitin cell wall. Marking attendance means liking the session. That is how I'll know. When you come to a class, they'll call, no, Lakshmi, are you present? So that time you'll tell, yes ma'am, I'm present. So like that you have to mark the attendance. Yes, very good. Fungi. Okay, next question. The only kingdom that can do chemosynthetic autotrophism. The only kingdom that can do chemosynthetic autotrophism. Fast, you have to type. Why is the chat slow? Fast. Which is the only kingdom that can do chemosynthetic autotrophism? Yes, only Pavitra's answer I can see right now. Others are still telling fungi. I think the chats are a little slow, or you people got confused. Very good. Yes, Subhashini and Pavitra has told the answer. Yes, Monera. Yes, Monera is the only kingdom that can do chemosynthetic autotrophism. Students, tell me the sole member of Monera. Who is like, I am it. Okay, I am Monera. Right? Who is the sole member of Monera? Who is capable of doing this chemosynthesis, autotrophism, heterotrophism, photosynthesis? Who are the people? Okay? Yes, give it a guess. Very good. Bacteria. Right? Bacteria. Not only Eubacteria, again you will write Archaebacteria and Eubacteria. Okay? Chalo. Understood this box very clearly? Okay? Here, peptidoglycan, do not forget this. And that's all. Very simple box. Okay? Simple box. Now, the next one that you have to study. Here, students, under your 5 kingdom classification, even though you understood the box, here and there some confusions are there. What do I mean by confusions? Some of them have like some algae were put into protista. They were supposed to be in the plantae, but they were put into protista. So let's see one very important paragraph of your NCERT. Most of the students ignore this paragraph and study the rest. Sometimes the question will come from this paragraph. Look here. Let us look at the 5 kingdom classification to understand the issues and consider consideration that influence the classification system. Earlier classification system include bacteria, blue-green algae, fungi, mosses, and ferns, gymnosperms and angiosperm under plants. So the first initial classification, whose classification they're talking about? Your Carl Linnaeus classification. All these classifications had all these people together under plants. But once who came? Whittaker came, he told no, this cannot happen, I will separate them. So he separated them. Okay, the characters that unified this whole kingdom was that all the organisms include cell wall in their cells. Okay, cell wall. One main thing that almost all the scientists took into consideration. This placed together groups which widely differed in other characteristics. As I told you, they're not mentioning clearly that this was based on Carl Linnaeus classification, but here you can see that they're telling, because you did a classification merely based on difference or presence or absence of cell wall, what happened? Organisms which were very far from each other also became relatives, and which were relatives got separated. Okay, so understood that. That is what they're trying to say. Very important paragraph of your NCERT. So please open. Okay. Okay, I will make you a way to remember this. Now, this is placed together groups which widely differed in other characters. It brought together prokaryotic bacteria and the blue-green algae with other groups which were eukaryotes. It also grouped, carefully listen, what are they trying to say? They categorized blue-green algae and prokaryotic bacteria together. Now, it also grouped together unicellular organisms and multicellular organisms. Whatever I told you about Carl Linnaeus drawbacks, it's mentioned here. Don't take tension. Okay, don't take tension. Fine. Now, most important thing is, imagine students, I'll write this. Okay, what they're telling here, I'm going to write it. Are you all with me on this? Yes, are you all with me on this? Yes, got it. Okay. So, according to Whittaker, your amoeba, okay, paramecium, okay, paramecium, same time, chlamydomonas, okay, chlamydomonas, spirogyra, no, spirogyra and Eulothrix I'll put separately, okay, chlamydomonas and your chlorella, only spirogyra is mentioned here. Cholera is not mentioned. Okay, cholera is not mentioned, but I am going to write it. Okay, I am going to write cholera because I have seen in some MCQs cholera will be mentioned. Okay. Yes. So students, here what happened? Your amoeba and paramecium. Okay, amoeba, paramecium, your chlamydomonas. They all, even though chlamydomonas and your chlorella came under algae. Okay, they are actually what? They are actually algae. Yes or no? Yes, they were supposed to come under plants. But according to Whittaker, he put them under Protista. He put them under Protista. Because Protista was for all the organisms which are eukaryote but unicellular, right? Unicellular eukaryotes were put under Protista. So he told, even though chlamydomonas and chlorella might fall under algae or have all the characteristics of algae and should be under algae, he was like, no. According to the classification, this will come under Protista. So only thing you have to remember is C square. C square is put into P. Okay, C square is put into P, that is CP, okay, CP. That is chlamydomonas, and again students, if you cannot remember chlorella, it's okay, at least chlamydomonas remember, because see, in NCERT it's mentioned very clearly, okay, very clearly. So this C was put under Protista, that is, it was supposed to go under algae, plantae, it was put under Protista. Yes, got it, clear or not? Yes or no, tell me. Is it confusing? Is it confusing? Then one more example, okay, one more example is your spirogyra and Eulothrix, okay. Where do I write it? Chalo, we'll write it here. Yes, spirogyra and Eulothrix. Okay, this category also was put under your plantae. So here you can see chlamydomonas and spirogyra were placed together under algae, but the classification did not differentiate between the heterotrophic group, that is fungi, the autotrophic green plants, though they also showed the characteristic difference in their wall composition. So Spirogyra and Eulothrix also, even though they are, you know, your multicellular green algae, they are now under your plant kingdom. Okay, they are under your plant kingdom. That's all you have to study. Okay, plant kingdom, they are green algae. Okay, got it? This is the only classification. Students, Chlamydomonas is the confusion. Okay, because Chlamydomonas is an algae, but you're studying it under Protista. Okay, that is the only confusion here. Nothing else is a confusion, but an important paragraph. Okay, little confusion can come while telling it. Okay, but otherwise there's no confusion. Is this clear? I have written what is mentioned in the paragraph. Again, even yeast also, even though it is coming under unicellular eukaryotes, it is in Protista, but you will study it under fungi. So that's all. That's the small variations that is there in this chapter. Because understand, some of it, you know, some of the people might behave like a fungi, but because again, like they are unicellular and they are eukaryotes, they are put under Protista. Is this clear? Can I get a thumbs up? Can I get a thumbs up if it's clear? Yes or no? So what is the problem? Chlamydomonas, even though it behaves, it is an algae, it should have gone under plant kingdom, but it is right now under Protista. Why? Because it is a unicellular eukaryotes. Clear? That is the only thing. Spirogyra and Eulothrix, you will study it under plant kingdom. Okay? Yes, correct. Okay, some more exceptions might be there, okay, because we do not read that much, no. Maybe now also many discoveries are happening and such kind of you know, changes would have happened, but as of now, according to NCERT, only Chlamydomonas is there. I told you Chlorella as well. Okay, yes. Students, now we are going into the classification. So the first ever kingdom was what? Kingdom Monera. So Kingdom Monera, the sole members of Kingdom Monera are none other than, who are the sole members? The sole members are your powerful bacteria. Okay, is there anyone who is as powerful as your bacteria? Not at all. Wherever you go, bacteria will be there. Okay, sole members are your bacteria. Okay, sole members are your bacteria. Now they are prokaryotes. Do anyone have doubt in that? Prokaryotes, again their cell wall is made up of peptidoglycan. Okay, peptidoglycan. Yes. Again, you will hear the word murin. Okay, you will hear the word murin. I'll tell you about it. Cell wall is made up of murin. Yes, peptidoglycan, or the other word is murin. Do not be like, oh, ma'am did not teach us this. This word also, instead of peptidoglycan, if you see murin anywhere, understand, again, they are the same words. Yes, got it? Clear or not? Yes, Lakshmi has too many doubts today. How long will be the session? What is that? What is this? Yes, okay. Murin, students, E is there, okay. Now, where do I find these people, right? Where do I find these people in this, where are the location of these people? They are found in all the extreme conditions. They are found in all extreme conditions. What do I mean by extreme condition? They are seen everywhere. Wherever there is extreme temperature, they are there. Okay, extreme heat, that means desert areas also they are there. Desert is there. Cold, snowy places also they are there. Okay, they are also there in extreme salty conditions. Every single place they are there. Okay. Oh, okay. Every place they are there, including, you won't believe, that is even inside our living organism. That means they are also present in plants and animals. Yes or no? Now if I touch this pen and if I go check under the microscope, I'll be able to see so many bacteria. Right? So many bacteria. There are good bacteria as well as the bad bacteria. Got it? Good bacteria as well as bad ones. Yes, exactly. Where only few life exists, those places your bacteria will be there. Why this bacteria is there, students? Please understand, it's because of their cell wall. Their cell wall is the one, the cell wall and the cell membrane is the one that is making them to survive any kind of extreme temperatures, extreme conditions, right? Yes, they are cosmopolitan in nature. Okay. Now, ubiquitous also in nature. Now, is this clear about kingdom Monera? Very general thing about kingdom Monera. Now, if I have to talk about classification, okay, classification of kingdom Monera. Chalo, let's go to the next page. Classification of kingdom Monera. Monera can be basically classified into three categories, I would say. So, I'll tell you why there are three categories. First category is your Eubacteria. No, let's go with Archaebacteria. Archaebacteria. Your textbook order will go Archaebacteria. Are you all going to read NCERT after this? Yes or no? Otherwise, no use, okay, of the class. First, I'll categorize them into Archaebacteria. Then, I will categorize them into Eubacteria. Okay. And then, there are some normal bacteria which you are seeing down. Okay. There are some normal bacteria that are living down here, okay, on the board. That is, this normal bacteria are basically classified depending on their shapes. Okay, so shall I write normal bacteria? I'll just write here normal bacteria, or I'll just write simple bacteria. That's more better than normal bacteria. Okay, simple bacteria. And this simple bacteria, students, they are basically classified based on their shape. Okay, classified based on their shape. The one that is in your textbook that you are going to see, based on shape. What are the shapes that are present? First one, you have cocci, that is your circular bacteria. Okay, spherical one. Spherical bacteria is there. Okay. Then you have your bacilli, which is rod shaped. Here it looks like spoon. I don't know why it looks like spoon. I have seen, I have done experiment with your rod shaped bacteria. I have done gram positive, gram negative. Okay. I'll start physics also. Yeah. Okay. Yes, based on shape. Okay. Yes, based on shape, what is happening? This is rod shape. Okay, mine is also not that rod shape, but students adjust. Okay, Gopika Ma'am, no, you adjust. This is rod shaped. You'll be doing all these experiments later. Okay, this is rod shaped bacteria. Again, I'm only using the simple terms. Okay, cocci, bacilli, you have to remember. Then you have your spirilla. Okay, spirilla. Students, yesterday I saw king cobra. Okay, when I was walking and going. Okay, spirilla. Spirilla. Got it? Yes. The last one is your comma shaped or your vibrio. Okay, vibrio. Okay, vibrio. Comma shaped. This is the common, depending on the shape. Okay, comma shaped. You have to know the scientific name. Scientific name, that is your spherical, rod, spirilla and comma shaped. Okay, comma shaped. Is this clear? So based on their shapes, this simple bacteria are classified. Now if I have to talk about Archaebacteria, this also is present in extreme conditions. Okay, they are also seen in extreme conditions. Again, they have false murin. Okay, false murin means like a false peptidoglycan, false cell wall. That is the reason why they are able to withstand any kind of extreme condition. Eubacteria simply means true bacteria. Okay, true bacteria means they have proper true cell wall. Okay, these people have pseudo. Okay, they are pseudo, false people, fake people. Okay, is this clear? Is this clear? Everything became yellow, no? Okay, now we'll start with Archaebacteria. Ready, steady, get set, go. Don't go, stay here only. Yes, get set, go. Done? Yes, done. Okay, so Archaebacteria, as I told you, this Archaebacteria can withstand extreme conditions. Okay, extreme conditions. So the first point is they can withstand enough of yellow, students, too much of yellow. Okay, they can withstand extreme conditions. Okay, What is the next thing? The most important thing is their cell wall is different and their cell membrane also is different. But cell wall here, I am going to tell you the name. Okay, cell wall is made up of, okay, cell wall is made up of, can you tell me? I just told you now, made up of pseudomurin. Okay, pseudomurin. What is pseudomurin? Basically a false peptidoglycan. Okay, false peptidoglycan. Yes, okay, fine. Now, the next thing about these people is they have a cell membrane. Students, they have a cell membrane that is also different. Again, this came as a PYQ. So, the question was, what makes Archaebacteria withstand in extreme condition? But the options made, the cell wall was not mentioned. So, students got confused. Please understand cell wall and cell membrane. Both of them are going to help these people to withstand extreme conditions. Okay. So, cell membrane is made up of monolipid. Okay. Monolipid, and this lipid layer is branched. Okay, this lipid layer is branched. Okay, easy, easy. So, please understand, not only cell wall, cell membrane also helps for this Archaebacteria to stand or withstand in that extreme conditions. Got it? Yes. Okay, yes. Okay, very good. Pseudomurin. Okay, the cell wall is branched and it's a monolipid. Now, which are the climatic conditions that they can withstand? Okay, some names are mentioned, some categories are mentioned, so we are going to understand that. So, if I have to write Archaebacteria, okay, categories, students tell me, Carl Views put Archaebacteria under which domain? Which domain? Let me see how many of you remember what I am teaching. Come on, which domain did he put them? Fancy colors and all I am trying. Okay, so Archaebacteria was divided into, come on, fast. Your fingers, your typing has reduced, speed has reduced. Why you people were on vacation, that's why. Halophils. Very good. Archae. Okay, archae. Monolipid layer, dear. Monolipid layer. This is how your lipid layer will be. It has a hydrophobic and a hydrophilic tail. Right, a monolipid layer. Okay, bilipid means like this. Okay, a plasma membrane is bilipid layer. Like this. This will be bilipid layer, that is 2 layers. Monolipid means single layer. Okay, it will have a hydrophilic and a hydrophobic head and tail. Okay, chalo. Halophils, that means extremely salty condition. Okay, extremely salty condition. Then you have thermoacidophils. Okay. They could have just kept Gopika name like this, but they didn't do that. They kept thermoacidophils. Next one is methanogens. Okay, methanogens. Now, tell me what is halophils. Let me see your chemistry knowledge. Halogens you study, no? Halophils mean extreme salty condition, like brine. Okay, extreme salty conditions. Now, thermoacidophils means extreme temperature, hot springs, even the acid pH can be very less. Okay, acid pH can be very less. That is thermoacidophils, hot springs. With that, even the pH can be low. Okay, pH low means extremely acidic. Okay, acidic. Okay, I am writing it here. Students will think pH low. What has this ma'am written? Okay, acidic condition. Okay, what is Vipul telling students? Oh God. Thank you. Yes, fine. Okay. They're talking to each other. Okay, so yes, next is methanogens. Okay, methanogens are, students, your seen in marshy areas. With marshy areas, you will study about this fellow in microbes. Okay, you will study about this fellow in ruminant animals. Gut of ruminant animals. Okay, gut of ruminant animals. Easy? Easy? Yes or no? Tell me, easy or not? Yes, very good. In the gut of ruminant animals. Who is your ruminant animals? Your cattles. Your cattles, right? They first swallow and then they bring it back and then they chew the food. So methanogens are there in their body. That is the reason why we are using gobar for forming your biogas. Got it? Because this can release methane. Methane gas can be released. That is why we are using that as the biogas. Okay, ruminant animals are the animals who, cattle students, all the cattles and all, their digestion is completely different, right? First they swallow the food, then they bring back the food and they aram se sit and chew. We do not have the time, so we chew, talk, we do everything, multitasking we are organisms. Is this clear? Is this clear? Is any student having doubt? You shouldn't have doubt, first of all. If you have doubt, okay, I will take it up. Yes, tell me, anyone doubt? Any doubts so far? Now only you can ask me doubts, okay? After I finish the class, don't ask doubts. Ma'am, you didn't tell that. Okay? Yes, tell me. Any doubts? Clear? Okay, shall I continue then? Okay, now we'll go into true bacteria. We are true people students, so we have to go to true bacteria. Okay, so extreme, understood this one, very simple. Extreme conditions, they can be seen, and cell wall is made up of pseudomurine. Okay, very important. Pseudomurine. Cell membrane is made up of a monolipid layer which is branched. Okay, monolipid layer which is branched. Again, Archaebacteria is divided into three, that is they are seen in extremely salty condition, extremely hot conditions, as well as acidic pH, and methanogens, that is in that is your marshy areas. Methanogens are present in marshy areas as well as in the gut of the ruminant animals. Do not forget your biogas. Now we are going to understand Eubacteria. Okay, Eubacteria or true bacteria. First ever thing about Eubacteria is true bacteria. That means they have, is the color visible? Yeah. Okay, true bacteria. What is this true bacteria? That is, they have presence of cell wall. New spelling I invented. Okay, so presence of cell wall. Presence of cell wall, that is your peptidoglycan layer. Okay, peptidoglycan layer. Okay, now let's understand the categories. Okay, Eubacteria, if I have to divide, Eubacteria. Chuma, I can't keep it, no? So if I divide Eubacteria, what are the categories that it will go into? Eubacteria can be divided mainly into two types. Okay, first one is your Cyanobacteria. Cyanobacteria, also called as blue-green algae. How many of you have heard this word blue-green algae? Tell me in the chats. Cyanobacteria and Mycoplasma. Okay, Mycoplasma. Clear? Cyanobacteria, blue-green algae. Did you hear ever in your life? Blue-green algae. Yes. Tell me fast. Blue-green algae. Yes, Cyanobacteria, blue-green algae. Okay, you have heard about it. Okay, let's talk about Cyanobacteria, students, first. Now, what do I talk about Cyanobacteria? I have to know how are these people. Like, are they there? Are they, filamentous? All that. So, Cyanobacteria, if I have to talk, okay. Cyanobacteria is also called as blue-green algae, BGA. Okay, blue-green algae. Again, you might have a confusion. Why this fellow is called algae, ma'am? You're confusing. You're more confusing than Whittaker. No, students, this is called blue-green algae because it has something like a plant or something like an algae. I'll tell you what is that something, but right now, don't take tension about that. Just understand what these people are. First of all, they are unicellular. Okay, they are unicellular. They are filamentous. Okay. Filamentous, and they also stay as colonies. Colonies means what? Have you seen colonies? Same set of people, maybe could be from a same culture, same caste, or from a same category, whatever they may be, they'll all stay in one area. We call it a colony, right? We call it a colony. So, BJ are not blue green in color. Okay, unicellular filamentous, and they will be colonial. Okay, colonial, that means they form colonies. Now the next important thing about this bacteria is, say they stay in colonies, okay, they also have a covering, they have a mucilaginous covering. All of you open your textbook, faster. They open your textbook and tell me if they have mentioned or they have, you know, marked mucilaginous covering. So these colonies are generally protected. Okay, this is basically for protection. Generally covered, we can tell, by mucilaginous sheath. Okay, mucilaginous sheath. Now you might be confused, how ma'am, how do they look like, right? So students, these are the colonies. Nostoc and all is given as your example. In between there will be some other cells. Normally all the cells, because they are autotrophic in nature, they can do chemosynthetic as well as photosynthetic. They will be doing carbon dioxide fixation. But in between there are some cells like this green color which I have drawn. No, color is not very visible. No, wait, I'll draw it with another color. Students, in notes all this will be full jatang colors, it will come out, but in the board it will not come. Okay, this green color cells which I have drawn, let's draw one more cell. Green color cells which I have drawn are the cells which will do nitrogen fixation. What will they do? Nitrogen fixation. Now that is not what I was talking about. I was talking about their colonies will be covered with a mucilaginous covering. Okay, mucilaginous covering. So look here, this is the mucilaginous covering. I am drawing every diagram of NCERT. Now if you people don't study, no, then I will scold you people. Then you will tell me scolding ma'am, strict ma'am. Okay, this pink color which I have done is a mucilaginous covering. Okay, are you all with me? Okay, mucilaginous means a gel-like covering, dear. Okay, a gel-like covering. Okay, a coating, right? So this pink color which I have drawn is basically my mucilaginous covering. One second, yes. Okay, mucilaginous sheath, covering, whatever you want to tell, you can tell. Okay, and what have I drawn? The green color. So normally a cell, what does it do? It has to do autotrophs. Okay, so they will take carbon dioxide and they will give oxygen out. Normal how a plant does photosynthesis, like that, these people will take light and form their own, make their own food. Now other cells are carbon dioxide fixation cells, but the cells which I have drawn in green are specialized cells which are called as heterocyst. What are they called? Heterocyst. Okay, heterocyst cell, main job for them is to do nitrogen fixation. Their main job is to do nitrogen fixation. Okay, clear or not? Okay, very good. Okay, main job is to do nitrogen fixation. So here you can see two heterogeneous cells I have drawn. Now this is a colony. Okay, this is a colony. Is this clear, students? Easy to understand? Any any difficulty? Now students, this other specialized characters that these people have is, they have presence of gas vacuoles. What do they have? Presence of gas vacuoles. These gas vacuoles are not there in all the organisms. Gas vacuoles are only present in the ones which are water blooming, you know, cyanobacteria. Okay, water blooming cyanobacteria. Now students, how many of you have gone to village? You would have all gone to village, you know. When you're going to village, you would have seen a pond which is filled with green color, right? If this is the pond, imagine this is the pond. One second. First of all, I don't know how to draw a pond. Okay, this is a pond. Okay, this is the pond. Now in that pond, when you're going, right, you would have seen like this. Entire pond is filled with one green color, like a very slimy layer. Okay, slimy layer. If you go there and if you, like you're some tourist or something, you will go and remove it from your hand, right? What is this actually? This is your algal bloom. Okay, algal bloom. Algal bloom. This people can cause, because I told you they have something like similar, like very similar to that of an algae or that of a plant. I'll tell you what that is. Okay, so this is algal bloom. Now what happens because of algal bloom is, students, down, okay, below this water, like here in the below, there will be lot of dissolved oxygen, right? Lot of dissolved oxygen will be there. All this oxygen will be consumed by these people to make their own food. All this oxygen will be consumed by these people to make their own food. Now because of that, what happens? The animals and the plants that are living in the bottom, right, living in the bottom, will not get any sunlight. At the same time, they will not get any oxygen and they will all end up dying. So algal bloom is actually bad, and many places, like if you see Kerala and all, many places have been covered with algal bloom because everyone are running behind forming city, like building, buildings, construction, everything is happening. So there is no time to take care of rivers and ponds like before. Okay, yes, got it. Okay, fine. So they have gas. Who has gas vacuoles? Water blooming algae have gas vacuoles to do the buoyancy. Okay, to do the buoyancy. So gas vacuoles are present in water blooming algae. Okay, that helps them in buoyancy. Okay, that helps them in buoyancy. I know some might come out as an additional information, but I would have seen this in PYQs or somewhere. When I read it, I realized that, okay, this is important for the students. I'm discussing that. Okay, chalo, understood? Okay, we have some more things about your Eubacteria, that is especially about your Cyanobacteria. Look here, I have mentioned it here. There are thousands of different Eubacteria or true bacteria that are characterized by the presence of the rigid cell wall or the true cell wall. And if motile, a flagellum, the Cyanobacteria, also referred as blue-green algae, have chlorophyll A. This is what I told you. Why did I call a Cyanobacteria blue-green algae? Is because Cyanobacterias, okay, because Cyanobacterias have the presence of your chlorophyll A. See, they have phycoerythrin and all that, but currently you need to know why are we calling them algae? Why are we not calling them anything? Because they have chlorophyll A in them. Okay, very important, chlorophyll A in them. Now look here, students, to green plants that are photosynthetic autotrophs. Cyanobacteria are unicellular, colonial, filamentous, and freshwater or marine or in terrestrial algae. Now the colonies are generally surrounded by a gelatinous sheath or a mucilaginous sheath. They often form algal blooms in polluted water areas. Some of these organisms can fix atmospheric nitrogen. The specialized cells are called heterocyst. Okay, heterocyst. Please don't talk with yourself. Please focus here. Talk to me. Okay, some of these organisms can fix atmospheric nitrogen in specialized cells called heterocyst. Nostoc and Anabina are one of the examples. Okay, Nostoc and Anabina. Now, chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites and ammonia and use this to release energy for ATP production. That's why I told you these are the only people that are capable of doing chemosynthesis. Okay, chemosynthesis. Is this clear? Yes. Yes, Vipul, clear. Pavitra, clear. Okay. Now, other thing about this Cyanobacteria, students, they are symbiotic in nature. Okay, they are symbiotic in nature. That means they have connections, right? They help another person and they also take help. So they will not harm each other. So that means they are positive and positive. This is science. Okay, symbiotic example you can remember: Anabina plus Azolla. Actually, Azolla is a fern. Okay, Azolla is a fern that will help again. Both of them they'll each other help for nitrogen fixation. Okay, yes, clear. Chalo. Okay, now blue green algae, why it is called? Because of the presence of chlorophyll A. Is this clear to all of you? Yes or no? Tell me fast. Okay, next thing we have to go into is Mycoplasma, students. Mycoplasma is very simple. Nothing is there actually to learn about it, but again I will mention it here anyways. Okay, your Mycoplasma is the next category. This Mycoplasma is one of the smallest living fossil, okay, or the smallest living cell, okay, living organism, smallest living one that is present. Here one exception is there. What is the exception? Can anyone tell me? What is the exceptions? We kept telling about cell wall, cell wall, cell wall. This fellow has no cell wall. Okay, no cell wall. So this Mycoplasma is little special. It's very special fellow. Okay, very good. No cell wall and they are pathogenic. That means they can cause disease very easily. Okay, pathogenic. Got it? Pathogenic, clear? Yes, smallest living cell is Mycoplasma. Okay, now they can also do, students, in the absence or presence of oxygen. In the sense, if oxygen is there, okay, I'll do aerobic. If oxygen is not there, I'll do anaerobic. That kind of organisms are called as facultative anaerobes. Okay, facultative anaerobes. Okay, that means depending, if oxygen is there, I'll do. That means if ma'am is teaching, I'll listen. If ma'am is not teaching, I will not listen. If you want, I will crack Neet, or if you don't want, I won't crack Neet. Like that kind of people is facultative Neet aspirants, right? So facultative anaerobes. Got it? Yes, they can do anaerobic respiration. Gram positive, gram negative, I'm not mentioning because you don't know the concept. If you do not know the concept, it will just be a word. You have to know what is this concept actually talking about. Chalo, now Mycoplasma is done. Mycoplasma is done. Now we'll learn a little bit about their reproduction and nutrition. Okay, reproduction and their nutrition. Ayo, why is this line coming with me? 1 second, students. Circus I'm doing. Yeah, so yes, let's do nutrition. Okay, what is the nutrition? Nutrition is very simple, students. I'm just writing it here separately because again you might miss out on the nutrition. Okay, 3 types. Let's do autotrophs. Okay, autotrophs or autotrophic. Okay, 1 second. Chemosynthetic and heterotrophic. Okay, chemosynthetic and heterotrophic. Heterotrophic. Now, what is autotrophic? Very simple, ma'am. They make their own food. Why are you making it a big deal, right? They make their own food. But under this, you also have phototrophic and chemosynthetic. Do not forget, I'm talking only about Monera. Under autotrophs, 2 things was mentioned. What were they? Chemosynthetic, okay? Chemosynthetic and photosynthetic. Photosynthetic. So, photosynthetic is not first time that you're hearing, but what you're hearing chemosynthetic for the first time. Yes or no? Yes or no? So, photoautotrophism. Okay, photoautotrophism. 1 second. I'll write photoautotrophism. That means using light. Okay, photoautotrophism. That means basically with the help of sunlight. Similar, students, there also we use sunlight only. Okay, with the help of sunlight, I make my own food. Yes or no? Easy? Yes, okay. With the help of sunlight, I make my own food. How am I possibly able to do it? Because of my pigments, right? Only if pigments are there, I will be able to do. That means because of the chlorophyll that is present. Okay, chlorophyll that is present, I am able to do this. Any doubts so far? Is this clear? Okay, how am I able to do photoautotrophism? Light is there. Okay, sunlight is there. But for that absorption, I need chlorophyll in my body. That is there in my Monera. Got it? Photoautotrophism. Now, when I have to talk about chemosynthetic, okay, chemosynthetic or your chemoautotrophism, okay, I think I'll write those words only, students, because I'm scared. What if we write some word and need NCRT? I mean, NTA gives you some word, then many students will leave it and come. Okay, autotrophism. Okay, because it's coming under autotrophism, we are writing chemoautotrophism, photoautotrophism. So that only I'll continue here also. Okay, that is chemoautotrophism. Okay, tropism. Now, chemoautotrophism means very simple. Like the before slide, it was already mentioned that these people use nitrates. Okay, nitrites, ammonia. They use all these people to finally form ATP, to do ATP production. Is this clear? That means I will use chemicals, right? I will use chemicals to make my own food and to yield energy out of it. Easy peasy? Yes or no? Okay. Is this clear? So that is basically, that means I am using nitrates, ammonia, utilizing all the chemicals, and because of that forming the ATP, the energy that is required for my body. Is this clear? Okay, they will oxidize the chemicals and they will yield out the energy. Okay, oxidize the chemicals. Oxidize the chemical to yield out energy. Now the last one is heterotrophic. Students, heterotrophic is not new for us. What and all have we learned at a heterotroph? You have saprophytic, parasitic, right? Saprophytic and parasitic. Okay, that is again decomposers, all of that. I'm not writing much about it. I'm just writing saprophytic. Okay, under saprophytic, please understand they are using the dead and decayed. They are utilizing the organic matter, right? They are utilizing the organic matter. Okay, so the saprophytes, basically they consume the dead and decayed. Dead and decayed. Decayed. After that we have parasitic. Okay, parasitic. That means they feed, they stay on the host body and consume whatever that the host is giving. It's like, okay, host is eating this, I will eat this. Now some examples are mentioned in your textbook, so I'm writing that. Tetanus, okay, the diseases caused because of it. Okay, diphtheria. Yes, and you can also write many other diseases are there, okay, but you can write few of them. And cholera. Cholera. I have very much confusion with, see now also confusion is chlorella and okay, whatever. Okay, students, so confusion. Some words are always like that. Okay, it does not just come out like that. These three they have given under parasitic. The last one is what? Under heterotrophs, the last one is what? Symbiotic relationship. You can write about rhizobium that is present on the root nodules. Okay, rhizobium that is present on the root nodules. Again, see, parasitic you can write many are there, but you only have to remember as of now tetanus, diphtheria and cholera. Okay, cholera. Okay, yes, clear, easy. Okay, last one is symbiotic relationship, that is mutualism. Okay, mutualism. Yes, lichen symbiotic relationship, but that is again fungi and algae. So here you have to remember rhizobium, which is present on the root nodules, and you can also remember azorhizobium, which is present on the stem nodules. Okay, stem nodules. Clear? Yes or no? Okay. Azorhizobium you can remember. You want me to write that? I'll write it on the top here because I don't want it to go to another page. Okay, I'm writing it here. Okay, last one is symbiosis, that is example rhizobium. Okay, rhizobium with the root nodules. Yes, done. Done. Clear? That's all. Next topic, last topic for the day is reproduction. After that we are done for this topic. Okay, the last topic is reproduction. How do these people reproduce, ma'am? They are telling that these people are not doing sexual reproduction, but the bacteria that used to die before with some kind of medicine is no more dying. Right, no more dying. So then the problem is, oh no, then I have to know something more, right? So these people, if I see the entire, you know, their reproduction, asexual reproduction and sexual reproduction, right? So here they are telling that the sexual reproduction is completely absent. Okay, sexual reproduction is completely absent. But please do understand, transformation happen, your transduction happens, all this happens. That means, like, transformation, students, your transforming principle, like your Griffith's experiment, something is getting transformed from one person to another, right? A small DNA transfer is happening. All that also changes the entire genetic characteristic of it, right? You have conjugation, transduction, transformation. So I'm going to write here sexual. Okay, no sexual phase. Okay, no sexual reproduction, or we can write sexual reproduction is absent. Okay, sexual reproduction is absent. Okay, is absent. Then ma'am, how are they changing? Is bacteria primitive now also? No. From the time bacteria has come, they have modified themselves. If sexual reproduction does not happen, then there is no variation that is going to happen. But how are they doing this? That is because of other methods like your transformation, okay, transformation, transduction, okay, transformation, transduction and conjugation. Okay, conjugation. You can read a lot about this. This is a topic by itself. Conjugation, transformation, transduction, but this much knowledge is enough because students will have question. Okay, students will have question like, oh ma'am, how are they doing this? They have modified themselves, but sexual reproduction is not there. That kind of confusion to avoid, I'm adding this information here. Okay, chalo, let's do sexual reproduction. I mean asexual reproduction. Okay, asexual reproduction is mentioned in your textbook. Can all of you tell me? Very simple. You have heard binary fission, right? So during favorable and unfavorable condition, they take different methods. You know that before also. Again, when you study your fungi, when you study your protista, you will read this word. You will see that during favorable condition they behave differently. During unfavorable condition they act differently. Is this clear? Yes. Reproduction without multiplication. Okay. Unfavorable. During unfavorable condition. Students, always during unfavorable condition, the easiest thing that every organism does, that is your spore formation. So spore formation, that means they form the spores and they release it during unfavorable condition, so they get scattered and they form. Okay, now favorable condition, they will do binary fission. Okay, favorable condition, they will do binary fission. Students, all this is exact lines of NCERT. So that's the reason why the notes is like this. Every word is exact line of NCERT. Is this clear? Okay, binary fission and your spore formation. This is your entire reproduction by itself. Okay, entire reproduction by itself. Okay, reproduction. Clear? Is this clear? Did we miss anything? Look at your NCERT and tell me if we missed anything. If we missed anything or everything is done and dusted. Okay, so students, the next, that is your protista and fungi, will be done in the next part, because protista by itself is very long and I don't want to take a lecture that is like for 4-5 hours. Now the students don't have that kind of energy. Okay, now they are like, oh, re-neet will happen, re-evaluation will happen, that will happen, this will happen, full. Okay. Yes, bacterium done with Gopika Ma'am. No, biological classification. Monera done. Okay, done. Okay, no dear, this chapter is not completed. Done. Okay, something special is coming. No, don't forget, something special is coming for all my YouTube students who are waiting for so long. So don't feel disappointed. Something big is coming. Okay, very soon. The wait is almost getting over, right? So students, look here, the entire notes is made. Okay, I have handwritten every single word of your NCERT. So now your only job is to study. Okay, those who joined late, you can see the notes and you will get this notes where will you get it? Wait, I'll tell you. Okay, all the notes, all the updates, you will get it. Those students who joined us newly, please understand, we have some set of things. Okay, all the notes, okay, quizzes, updates, okay, updates, any changes in the YouTube class, all of this you will get it on Vedantu Neet English Telegram. Okay, where is the link? In the description. Neet English Telegram. Got it? And if you want to follow me personally, that is, you want to get all my classes, you want to have live conversation chatting with me, okay, then you will have to join. That is, for my thing you have to join Gopika, that is Biology by Gopika. Okay, Biology by Gopika. This is for all my updates, all my videos, all my live sessions. I usually send some motivation in between. All that will be here. Okay, these are the Telegram links for you to join. By this, you will always stay updated. Okay, you will always stay updated. Telegram channels to join. Now, other than that, many students will be having a problem that, ma'am, what to do next? Many students are asking, will re-NEET happen? Will re-evaluation happen? What am I supposed to do? My marks are here. What am I supposed to do? Then you will have a free one-on-one mentorship with Shreyas sir to clear all your NEET related doubts. Okay, that means many students will be there who will be in 400 to 550 range, and who really don't want to take a drop but want to do something else. Sir will give you, or we all of us will give you an idea about what you should do, what you should not do. By joining this, you also get exciting scholarship for your offline droppers batch, and also you will know whether to take a drop or not. So if you're a student who is still, because see, again for counseling we have some more time, and also for the Supreme Court to give the final verdict, right? For that also we have some more time, that is 8th July. So till that you might have a lot of doubts. If you have any doubts, students, go to the description box. You will be able to see the helpline number. Helpline number you can join and call and talk to. Not only call, you can connect with the entire our team. Okay, they will connect with you and they will tell you what you should do. Okay, lot of counselors are here. Lot of counselors are here. So they will talk to you, they will mentor you, and they will let you know what to do. I also hope all of you know about the DNA batch. Okay, something more exciting is coming there also. So DNA batch students who are in the class should definitely tell about how the DNA batches, because their students, the classes happen very slowly. We teach every single word in depth. Here also we teach, but they're little more slow we can do. On YouTube we cannot go like that. YouTube will never promote a video which is very slow. Okay, so in YouTube we have to be a little more faster. According to the YouTube statistics we have to work, but there it is our own app. So we can work according to our comfort. Classes are scheduled up to December. That means up to November, all the classes are scheduled and all the portions will be completed. Okay, chalo, make your decision wisely. Don't be late. And also NEETpedia, I hope all of you already know. Yesterday sir also mentioned, go and download the mind maps. This is my biggest thing. Molecular basis mind map I saw and my mind blew. Okay, so yes, thank you so much for joining, students. The next part, that is your protista and your fungi, will be taken in the next class again. So let me know how was the classes in the comments. If it is your first class, definitely comment down. Tell ma'am it's my first class, my name is so and so, and how was the class. Okay, because feedbacks matters. Positive criticism always helps us to become better. Okay, thank you so much for joining. Take care all of you. Stay hydrated, stay happy. Everything is going to be fine. Okay, and continue your preparation. Do not stop it anywhere. Okay, we are with you. I am especially with you. So yes, okay, so do not forget to like this session, share this video, also comment. Okay, some of you are commenting every time, some of you are not commenting. I am watching you people. Okay, yes, punishment will be given. Tata, bye bye.